Hi everybody, welcome back to our new Bio 107 virtual session. So this is going to be our new normal here for a while, unfortunately. But we're going to make this work, and hopefully it won't be much different than what you're used to. At least that is the hope. So the format's going to be the same. The material is all going to be the same, and we'll just try to make do with what we have. So during Module 8, we're starting that up now. We're going to be talking about speciation and extinction. So you can find your readings here on pages 128 to pages 145. And so you'll be able to do your readings like normal, and hopefully it won't be that much different than what you would have for your module, for your quizzes, or anything else. So we're making it work. Okay, here are just important updates. So spring break, we all know, was extended by a week. And so now the new official start date back from spring break is March 31st. This was just to give a lot of the instructors time to quickly convert all of their lectures to a virtual format, and also prepare for the conversion for the rest of the semester into an online learning, e-module, whatever they're doing. I guess it varies depending on what sort of class that you're in. I just want to point out that some of our due dates have changed. So I have updated for the next 2 weeks in our Cobra the calendar. So just be sure to take a look at that. There are some dates that are different. Some dates are still the same, but it has altered and altered the rest of our semester slightly. So just be sure to check your calendar on Cobra, and that way you won't get too lost. And as long as you're watching the lectures, I'm going to be updating everybody on when things are due and what we're covering each week. Okay, so from here on out, all of our lectures are going to be recorded, and we'll put them on YouTube. And the link for the week, or I guess all 4 links for the YouTube videos for our 4 sessions per week, will be sent out via email. And so be sure to check your email for those YouTube links. Hopefully everybody will be able to watch YouTube either on their phone or on their desktop or iPad or whatever you have. So all of these lectures will now look like this, including our laboratory. Our labs themselves will also be virtual. This is going to be a little bit more difficult, but since a whole bunch of them aren't going to be super hands-on, we will be able to make this work. At least that's the hope. Or if we can't make a certain lab work, then we can find a suitable substitute for it. So going forward, all of our module quizzes will be done online in your homes. The testing center won't be open, so don't try to go to Parkland and use the testing center. So I'm taking off the Respondus lockdown browser on the modules quizzes, and so you'll be able to click and log on to that module quiz from your own device, and then you can take the quiz. The only difference is that it will be timed for 20 total minutes, and so only 10 questions. You'll be able to have 20 minutes to take the quiz, but the quiz itself is open book. You can use your notes, you can use the book, you can use whatever resources you'd like for those quizzes moving forward. Also, just watch your email for important updates. I know I have been kind of quiet lately in the past, but that was primarily because all of the lectures were being prepared for you guys. Okay, so last week's reminders. So these are some new due dates. So the week we last met back, I believe the second week of March, before the first official week of spring break, These are the reminders that you have still due, so please make sure you do this because a lot of you were in class and paid attention. So Module 7 quiz, that is going to be due Tuesday, March 31st. So make sure you do that. There's just only 10 questions. There are 10 random questions out of a possible 25 questions. Remember, it's open book, 20 minutes. And that's all about microevolution. So we talked about microevolution. We talked about different types of selection. I even sent out Friday's PowerPoint presentation to a lot of folks because I knew they weren't going to be in class. And things had changed. So just be sure to look for that email with that presentation in there as well. Also, lab number 7, the Hardy-Weinberg lab, if you didn't turn it in to me, that is going to be due Tuesday, March 31st. So you can turn it in 2 different ways. You can either take a photo of the front and the back of the worksheet that you all received, or you can scan it and send it to me, and I will accept it either via email, kbuckley@parkland.edu, or you can put it in Dropbox under lab number 7. So that's your Hardy-Weinberg assignment. And lastly, this is just coming down the pipeline so you can be aware this due date has changed. It was due a little bit earlier, but we've moved it back since you guys had the second week of spring break. The Bio News File, the third and last Bio News File, is going to be due Friday on April 7th. Actually, it's Tuesday, April 7th, but I'll change it before I submit it. So it's going to be Tuesday, April 7th, not Friday. It was due on a Friday, but now it's just due on Tuesday. Okay, so for this coming week, we have new due dates for this coming week as well. Module number 8, which is what we're going to be going over this week, that quiz is going to be due on Tuesday, April 7th. 10 random questions out of a possible 29 questions. Open book, 20 minutes. Also, lab number 8, which we are doing tomorrow virtually, we're doing island biogeography. So that is going to be due on Tuesday, April 7th as well. So keep that in mind. And then lastly here, again, Bio News File number 3, it's due Tuesday, April 7th, not Friday. It's April 7th is a Tuesday. Okay, so some of you also have questions about oral presentations. We are still doing oral presentations, but it's going to look a little bit different. For those of you who were not in class on the last Friday that we met, the oral presentations need to be 3 to 5 minutes long. Okay, now instead of performing them live in front of the classroom like we had originally intended during one of our lab sessions, you're going to have to record your presentations. You can do that with a cell phone or a webcam or whatever device you want, a GoPro, or you can send me your PowerPoint presentation and then you can present it to me live over the phone. So that's the less ideal option. However, I know some people don't have the technology to be able to record their presentation live as they're giving it. Okay, so those presentations are going to be due to me by April 21st. So that is going to be during one of our lab days, so you won't have an official lab that particular day. You will just have to turn in your presentation to me that day. Now you need to pick a topic for your oral presentation. So that oral presentation topic has to do with biological evolution. So you want to pick a topic that you're interested in, that we covered possibly during the course, or that we plan to cover during the course. And you have to select your topic by April 10th. So some examples of topics include maybe there was an article that you found on the internet or in a magazine. You want to explore a case study more carefully. That could be an option. Or if there's an example of evolution in action in a particular organism that you really like, that you'd like to learn more about, you can pick a particular organism, but it has to be related to biological evolution. So just make sure you're really tying that in there. So they're just some examples of topics. Now, once you have selected your topic for your PowerPoint presentation, you need to post it. Now, I don't want everyone doing the same topic, and so you have to post it under Module 8 in the forum. There is a PowerPoint discussion topic. So make sure to check out other posts of students who have posted there to make sure that you are doing something different. So just post right in the thread what your topic is, and you can be as specific as you'd like. Okay, for Module 8, so we're starting Module 8 today. So this is your Module 8 reading. In your book, it's going to be pages 128 to pages 145. And these are the 6 sections that we're covering. What is a species, different reproductive barriers, spatial patterns, speciation, extinction, and biological classification systems. Now, we covered what a species is in the past a little bit. We covered some speciation, but we're going to talk more about spatial patterns and reproductive barriers, and also different types of classification systems. Okay, so let's go ahead and get started. So we're covering the first 2 topics today. What is a species, and reproductive barriers. So you guys probably already have a little bit of an idea of what a species is. So when you're thinking of a species, I want you to consider how many species actually exist on Earth. So scientists believe that there's around roughly 8.7 million different species on Earth, but we've only been able to identify and describe about 1.7 million species. So there's a lot of different types of organisms out there. So how did they all get here on Earth? So we know that the fixity of species is something that was used in the past to describe how species came to be, whereas they all just existed and never changed throughout the course of time. But we know that that's not true. We know that they just didn't pop into existence because there's evidence. There's geographic evidence, there's geologic evidence, and there's other forms of evidence. So I want you to also remember how we defined species in the past, okay? So in the past, our best definition that we used for a species is a group of closely related organisms that can do 2 different things. The first is that they can interbreed, and the second thing is that from that breeding, they can produce fertile offspring. So you see in my image here, you have two different toads and 2 different frogs, species A and species B, frog species and a toad species. They produce offspring. They produce a toad offspring and a frog offspring. However, those 2 distinct species cannot produce a fertile or viable offspring. And if they do produce an offspring, it will be an infertile hybrid. So, and that infertile hybrid won't be able to reproduce, and basically that fitness for that infertile hybrid is 0. But we also know a few other things about a species. So we know that in general, a population of individuals that share an ancestral lineage is what we would call a species. And so this can get a little bit tricky because if you go back far enough, basically you'll see that everything is related depending on how far back you look. But here you can see that we have different mink species and different badger species. So how do you know where that line is drawn? We know that mink and badger are closely related to an extent, but where does that line get drawn? So that's what scientists have been trying to do for decades, trying to make sure that they differentiate well enough to classify them accordingly. Now, species come to be in a few different ways. I know in the past we've talked about macroevolution and we've talked about microevolution. So macroevolution is describing these very large and complex changes in all of life, and it tends to span very long periods of time, millions of years. Now, microevolution, however, is a process that happens fairly rapidly. And sometimes we can observe changes that have occurred in a particular species over just a few years. And typically you find microevolution occurring in species that reproduce quickly and have shorter lifespans. But it really depends on what you're talking about. For example, you might be able to see microevolution happening more readily in different mosquito species, right? So all of these mosquito species here are all different species because they're classified differently based on some different morphology or some different geographic or geologic tendencies. But depending on which particular species you're looking at here, it basically is trying to show you that all of these different mosquitoes are closely related, but in some shape or form microevolution has occurred. Now we also have species that don't necessarily look very much alike. In this instance, we have several species of lubber grasshopper, which is a large grasshopper that these two species here exist in the southernmost parts of the United States. And we have one species here that on the far left is a desert species of grasshopper, and the other 4 species are more of a wetland marsh species. They like humidity and wet areas. Now, the ones that exist in the humid and wet areas are more closely related, even though they look very different morphologically. Whereas the desert grasshopper, even though it could be essentially paired in with the rest of them, it is a completely different species based on its behavior and its habitat and a few other things. So it's really, it can get a little tricky when scientists are trying to classify different species based on certain characteristics. But over time, you'll learn in the next lab here tomorrow that there are 4 different ways we can try to better classify an organism, and that is morphologically, geologically, geographically, and genetically. Now, I talk about these scientists that have classified all of these different organisms, but who was the first scientist that started classifying all of these different organisms? Well, this was Carl Linnaeus. So his Latin given name was Carolus Linnaeus. So he was around in the 1700s, 1707 to 1778, and he's really known as the father of modern taxonomy. So he was a Swedish botanist, and he was one of the first individuals to classify life based on appearance. And this was a little bit tricky, but we do use some of his systems today. And he defined species as all examples of creatures that were alike in minute detail of body structure. And in addition to that, he devised a hierarchical system for classifying species. But one of the issues with his classification system is they did not take into account any evolutionary relationships. So his species weren't appearing or disappearing, and they weren't related to one another because he existed and lived before Charles Darwin came out with his principles and theory of evolution. And so his system, although rudimentary, because it didn't take into account that evolution component, it is still used today because it did make a lot of sense. So he developed the uniform naming system for naming species. And so this is called the binomial nomenclature. So this is the system of using 2 different terms to name a species of living organism. Now the first word or the first term is going to be your genus. And the second word or the term is going to indicate the specific epithet. And so in order to name a species, you need these two terms or two words. But there's some different components when using these two terms. You have to write them in a certain way. So the entire two-part name must be written in italics, so it needs to be italicized. Then the second thing is that the genus name is always written first. You always write the genus first. And along with that, the genus name always must be capitalized. So when we, humans, Homo sapien, the homo, the H, the genus in that term is capitalized. And then lastly, the specific epithet is never capitalized. So sapien, which is us, we don't capitalize the S in that. So when we talk about Homo sapiens, capital H for homo, and then sapien, lowercase s, but the whole word is italicized. So this is just an example of how it would look if you were naming a species. So this is our common bobcat. The scientific name for the bobcat is Lynx rufus. So we have the genus, which is lynx. You can see that the L is capitalized and genus comes first. And then you have rufus, which is your specific epithet, lowercase r in rufus, but it comes second. And the whole word itself is italicized. So make sure you know how to write a species out, or the binomial nomenclature system, how you write that out. So one other thing too, when selecting out and classifying species into distinct species, is that it was a little bit tricky for early scientists at first, especially when it comes to birds. A lot of times their feathers change from spring to fall, and even in summer and winter. And so sometimes birds were originally classified as two different species, whereas they were the same bird, but just because their feathers looked different in different parts of the year. So in this case, if you've ever tried to identify or look up, or avid bird watchers, warblers are one of the most difficult type of bird to identify and to classify, because they are very similar, but you can see that most of these warblers here in this image are different species. The northern warbler, the yellow warbler, Cape May, the pine warbler, the ruby-crowned kinglet, they're all different warbler species. Now there are some here, you can see it, for example, that some fall warblers look a lot like other species, or they look very different in the fall versus in the spring. But a species can be defined based on the potential to interbreed and produce fertile offspring. We know that. And so this is going to be called our biological species concept. Basically, this is saying that a species is a population that exists, or it's a group of populations, and those members can interbreed and produce fertile offspring. Now, one thing about this concept itself is that it does not rely on physical appearance. So you have, in this example here, our eastern and our western meadowlark. Although they look very similar, a layperson could even say that they are the same species. This is not true, because that concept does not rely on the physical appearance. It does rely on the fact that those members can interbreed. The western only can interbreed with western, and the eastern can only interbreed with eastern. But they can interbreed and produce fertile offspring, because these two different species have different songs, they have different behaviors, and they cannot breed with one another. But there are a few problems with the biological species concept, because there are a few places where it cannot apply. So it cannot apply to asexually reproducing organisms such as fungi or protists or bacteria. So it can't apply, that concept itself can't apply to those organisms. And it's also impossible to apply that concept to extinct organisms because we don't know really about potential interbreeding, fertile offspring, behaviors, anything. We don't know anything about the extinct organisms other than the remains in which we find and what can be drawn from those. The third thing here is that some types of organisms have the potential to interbreed in captivity, but they don't do so in nature. And this is where we talked a little bit about subspecies. For example, how we talked about the northern water snake and the Lake Erie water snake, how they're very different and it's a subspecies. Now, the issue with the subspecies here is that they're separated and typically will not interbreed. Although if they were put together, they could produce offspring, but they don't do that typically in nature. The same thing with the Sumatran and the Bengal tiger. They're separated geographically. And although they could interbreed, in nature they wouldn't do so. But if you had them in captivity, certainly they could breed. So the biological species concept does not take that into account either. And then lastly, reproductive isolation is not always absolute. So you may have closely related plants that may produce fertile offspring occasionally, but the gene pools themselves remain mostly separate. So in nature, that reproductive isolation of certain plant species is not 100% always. So just keep those things in mind, that's where the biological species concept cannot apply directly. And if you remember also going back a couple modules ago, speciation was something that we talked about. So speciation is the formation of a new species, and we see that happen when we have members of a population and they're no longer successfully interbreeding with the rest of the group. And we saw that happen with Darwin's finches. So we have an ancestor finch, and natural selection occurs, and from that we have speciation. We have 3 different bird species: an insect-eating species, a woodpecker-type species, and a seed-eating species. And that happens through a few different channels. It will happen through mutations, it will happen through natural selection, and it will also happen through genetic drift, just to name a few. So when we talk about these species and these different species forming and speciation, we know that there are reproductive barriers that cause this to happen, that cause different species to diverge from one another. So we're going to talk a little bit about reproductive barriers today. There are 2 overarching concepts when it comes to reproductive barriers. The first one is our pre-zygotic reproductive barrier, and the second one is our post-zygotic reproductive barrier. So that's pretty straightforward. Pre-zygotic is a barrier that's preventing the formation of a zygote or a fertilized egg. And this is the barrier we see most common. It's the prevention of an egg and a sperm ever even meeting in the first place. So reproduction is not happening. And there's a couple subcategories that we'll go over. So for post-zygotic reproductive barriers, that is the barrier that is reducing the fitness of a hybrid offspring. So reproduction has happened, a zygote has been formed and been created. However, there's an issue typically that's happening with the offspring in that particular instance. And so just keep that in mind. Those are the 2 overarching umbrella barriers that we have. So we're going to go a little bit deeper into each of those barriers. So when it comes to pre-zygotic reproductive barriers, remember, before the egg and sperm even meet, there's 5 different very distinct barriers in the pre-zygotic category. So we have habitat isolation, we have temporal isolation, we have behavioral isolation, mechanical isolation, and gametic isolation. So we're going to go through each of these 5 in a little bit more detail here. So the first pre-zygotic reproductive barrier is our habitat isolation. So this is basically saying that these individuals are experiencing this prezygotic reproductive barrier because they are living in different environments. So in this case, we have ladybugs here, and they feed on different plants, and so therefore they're not even on the same plant to begin with, and so their habitat is different, and they're not meeting, and they're not reproducing. So that is the first barrier. The second barrier here is our temporal isolation barrier, or our reproductive barrier. So this is when we see different growth, we see different biology, we see different activity. So I've included a couple different crickets here. So in an example with field crickets would be that field crickets mature at different times, different types of field crickets. And so therefore they are not meeting to reproduce, because when one of them might be mature, the other one might just be a juvenile and not ready to reproduce at that point in time. But you also see this happening when you have organisms that are active at different times of the day. You might have one that's active at dawn and one that's active at dusk. And so that can be an issue that's preventing that barrier, or that preventing that reproduction. So that is one of the prezygotic reproductive barriers as well. So that's just basically any activity that's happening, or any sort of bodily function that is preventing those individual organisms to meet and reproduce. Next on the list, we have behavioral isolation. So this is the type of isolation that's separating organisms or species based on different courtship activities. So we see this a lot of times with birds. Different species of birds have different ways of courting one another. Some of them might dance, some of them might sing a certain song, some of them might fluff their feathers a certain way. But those different behaviors for reproduction are very different. We also see this with frogs occasionally. Different types of frogs have different mating calls, and so those are different, and that is a call that one particular organism or species won't answer at all. So if the call is not right, then those species aren't meeting, they're not reproducing. So that is our prezygotic reproductive barrier as well. Okay, we also have mechanical isolation. So that is when our mating organs are basically incompatible with one another. And so you see this occasionally with different plant species. So some plants spread their pollen in certain different ways. So for example, one sage plant species might spread their pollen through bumblebees or bee species, native bees, whereas another sage species might spread their pollen through the use of a hummingbird, where another species might use a bat, or another species might use a type of moth or butterfly. So depending on how their pollen is situated and who's using the nectar and grabbing the pollen that is basically making the reproductive organs incompatible with one another, because they're using different venues or different avenues of spreading their pollen from one plant to another. A bee is not going to use the same flowers that a hummingbird will use, and so they're using completely different ways of spreading their pollen. Okay, and then lastly here we have gametic isolation. So basically this is happening when the gametes themselves cannot unite. And so we do see this. For example, I have included a photo here of red and purple sea urchins. And so when red and purple sea urchins breed, since they're non-mobile, they release their egg and their sperm into the water. But unfortunately, when it comes to red and purple sea urchins, their gametes are not compatible. The sperm and the egg do not recognize one another if it's purple and if it's red. Now, two purple sea urchin gametes will recognize one another and be able to reproduce, and two red sea urchin gametes will be able to recognize one another and reproduce, but the purple and the red can't do that. Their egg and sperm are completely different, and they will not recognize each other to create a zygote. So that is one of our other reproductive barriers here. So those are our 5 prezygotic reproductive barriers, and those are just the 5 different types of isolation that we have that is preventing our zygote from even being formed, and it's preventing reproduction from happening. And this is how species typically will stay separate in separate gene pools. These 5 different types of isolation encourages the separate species to only breed within their species. Okay, so we're going to move on to, for the next couple minutes here, our post-zygotic reproductive barriers. So we only have 3 of these post-zygotic reproductive barriers. We have hybrid inviability, hybrid infertility or sterility, and we also have hybrid breakdown. And so we're going to go through all 3 of these and talk a little bit about the different types. So if you remember, post-zygotic reproductive barriers are barriers that have happened after the zygote has been formed, after the organism has been born or created. And now that that individual organism is alive, there's going to be some issues. So let's talk about them. So the first one is hybrid inviability. So basically what's happening here is a hybrid offspring is failing to reach maturity. And a good example of this would be a eucalyptus tree. So you have 2 different eucalyptus species, and they are somehow able to create zygotes and then therefore seeds. But the issue with these seeds is, even though these seeds are created from these 2 different species, you have this hybrid eucalyptus seed now. The problem with it now is that it will not grow to reach maturity. It won't sprout. The seed exists, but it's not going to grow. It just is there, and that is basically where it essentially is dying. It's dying as a seed. It's just not going to grow. That's where the life of that seed is ending. It's not reaching any sort of maturity. It's not sprouting, not growing into any sort of eucalyptus tree or plant. And so that is the first post-zygotic reproductive barrier that we'll see, is that even though that zygote and that organism is created, it just doesn't go anywhere. Okay, so the second post-zygotic reproductive barrier that we're going to look at here is our hybrid infertility, or our sterile organisms. So we saw this when we talked about ligers and mules. So basically what's happening here is that a hybrid offspring is unable to reproduce. So it's able to grow into an adult and reach maturity. However, that offspring itself is unable to create offspring. And so when we have a liger, which is a male lion and a female tiger, they are able to reproduce and create a hybrid. However, this particular hybrid here in this image is unable to breed and create offspring. We see that in mules as well. A combination of a donkey and a horse creates a mule, and the mules are really useful for humans, and they're good workers, but they're unable to reproduce. They are sterile. And so this is one barrier that's preventing these 2 species from interbreeding with each other, because once they do breed with one another, that species, the new species that has been created, does not reproduce and is unable to go forward as a separate species. Okay, and so then our very last post-zygotic reproductive barrier here is our hybrid breakdown. So what's happening here is when we have a second generation hybrid offspring that has reduced fitness. Okay, so let's talk about this a little bit. So, okay, you have a hybrid here. You have a second generation hybrid offspring, and now you have this reduced fitness. So basically, mosquitoes, in this instant, we saw that image of all those different types of mosquitoes. They're all different species. those mosquitoes, if they ever have the opportunity to interbreed with another, they create a hybrid mosquito. So this offspring, this hybrid mosquito, can produce offspring, but they have offspring with these genetic abnormalities. So, for example, they'll have abnormal genitalia, and then they can't reproduce because unfortunately it wouldn't match up with anything that exists in any of the other species. And so then that is where the particular individual would then not be able to procreate going forward. So it would cease to exist there. So yeah, unfortunately, they won't be able to reproduce. Even after they reproduce, they have reduced fitness, unable to create more offspring. Okay, so that is our 3 post-zygotic reproductive barriers that I had to talk about today. So I'm going to end the lecture, the first online virtual lecture here. And the next one that you'll see, we'll talk about part 2. And we're going to be talking about spatial patterns and speciation. So please, if you guys have any questions or if something's not clear, look at your book, send me an email. I'll be able to be reached that way. My office hours are basically virtual at this point, so if you have any questions about any grades or anything that we covered or something was unclear, just feel free to reach out to me and let me know. I will be getting all of our grades updated and following up with you all soon.